For our presentation, our group researched the pulse chase assay. Let's start by going over what the purpose of this assay is in the context of cell biology. So, in essence, pulse chase experiments are really useful for tracking changes in the intracellular location of proteins or the modification of a protein or metabolite over time. In a way, you could think of it as GPS for a protein within the cell. If you know exactly where the protein is throughout given intervals of time, you could essentially connect the dots and create a path of its intracellular movement. Likewise, if you know where a specific protein is, you can know whether it is being modified or attached to other molecules. One of the most significant examples of how this research has been conducted is with the secretory pathway. But before we go into detail on how this unfolded, it's important to have a working understanding of how this assay works. So the pulse chase assay is split up into two phases, the pulse and the chase. During the pulse, a cell sample is exposed to a radiolabeled compound that will be incorporated into the molecule of interest. Often when tracking proteins, this involves radiolabeled leucine. The result is that virtually all proteins synthesized during this phase will be radiolabeled, granted that they contain leucine. During the chase, the cell sample is exposed to a large amount of the non-radiolabeled compound. Now, the purpose of this is to ensure that any leftover radiolabeled compound is washed away and diluted. In other words, if the pulse phase involved using radiolabeled leucine, the chase phase would use normal non-labeled leucine. From this point on, the protein synthesized will not be radiolabeled. So what's the purpose of this? Well, essentially what we're doing is we've labeled all the proteins at a specific point in time, and only proteins synthesized during the pulse period will be radiolabeled. Because we can make the distinction between the radiolabeled and non-radiolabeled proteins visually, we can track their movement over time. Okay, let's take a look at a very specific example of the pulse chase assay in action to reinforce this idea. This assay was actually used to determine the secretory pathway. When the research was conducted, the pathway was not very well understood. Researchers studied pancreatic cells, which were specialized for secreting digestive enzymes in the small intestine, and as a result, they were packed with rough ER and Golgi. So the experimental procedure involved a pulse period with radiolabeled leucine, followed by a chase with non-labeled leucine. This is what's being shown in Figure A. You can see the radiolabeled leucine indicated by the orange liquid added by the pulse phase. Immediately after, the chase with non-labeled leucine, which in this case is yellow, was used to flush any leftover radiolabeled material out. The idea here, as mentioned before, is that this allows the location of the proteins to be visualized over time, as only proteins synthesized during the pulse phase are visualized. The researchers then analyzed the cells at various points in time using audio radiography and electron microscopy. The radiolabeled proteins could be visually observed at these times. Their results have been summarized into Figure B. This allows us to see where the majority of the proteins are X amount of time after the pulse period. As a result, the researchers were able to conclude that the proteins must go from the ER to the Golgi to the secretory vesicles.
Hello, this is Aaron. So, to aid our understanding, we can visualize the steps of a pulse chase experiment as if we were using Google Maps to make our commute. Getting a phone with a GPS is our radio labeling of our molecule, the driver. When the satellites connect with the phone and display our real-time location to us, it mirrors the observing of radiolabeled proteins. Much in the same way that we were able to determine the secretory pathway of proteins, the AI built into Google can, over time, determine what your regular commute is. Better yet, the Maps AI can determine when there is traffic by tracking many drivers, reflecting how pulse chase can reveal when and where molecular interactions are happening in our target cells.
Hello, I'm Brianna, and I'm going to talk a little bit about our paper. The paper we chose was titled Investigating MHC Class 1 Folding and Trafficking with Pulse Chase Experiments. MHC stands for Major Histocompatibility Complex. MHC Class 1 proteins are found in the cell surface as integral membrane proteins. They're expressed on all nucleated cells and platelets. The purpose of these proteins is to display intracellular proteins to cytotoxic T cells. These cells kill certain cells that are damaged or dangerous within the body. Therefore, MHC class 1 molecules play a huge role in the immune system. This paper discusses how the folding, peptide binding, peptide optimization, and surface transport of the MHC class 1 molecules have been studied for so many years. There are many questions about MHC proteins that have been discovered using this method, but this article touches on how the pulse chase assay can determine information about these proteins. The main question this experiment is asking is how can transport and cleavage by endo-H of the MHC class 1 protein be studied with the pulse chase assay? The hypothesis of this experiment is if the pulse chase assay is used on the MHC class 1 proteins in conjunction with gel electrophoresis, it will be possible to determine the timing of its transport and degradation within and through the cell. Moving on to how the pulse chase assay was used to address the question. Cell lines are used to run the pulse chase assay because while primary cells are preferred, it isn't very common practice yet. The labeling time must be appropriate in order for initial maturation and transport rates to be observed. They say around 5 to 10 minutes of the pulse stage should be good. In this experiment, cells were metabolically pulse labeled with sulfur 35 methionine and cysteine. They were then chased with unlabeled methionine and cysteine. When this experiment was conducted, MHC class 1 proteins were endoglycosidase H treated, or endo-H. Endo-H is basically an enzyme that cleaves off certain glycans. However, at a certain point during the secretory pathway, the MHC molecules become resistant to in vitro treatment of endo-H. It was thought that this occurs within the Golgi apparatus. This means that when observing a gel electrophoresis, the proteins in the earlier stages before the Golgi will appear different than those in the later stages of the pathway. The glycans will be removed with the endo-H sensitive molecules in the beginning of the pathway, and the glycans will still remain with the endo-H resistant molecules towards the end of the pathway. Besides this, folding and degradation will also affect the bands in the pulse chase. The pulse chase assay in this experiment helped determine the rate at which transport was occurring when folding is happening and where degradation is happening for MHC class 1 proteins. We're able to see where the labeled virus unlabeled molecules are versus unlabeled molecules are and how they are behaving throughout this process.
Hi everyone, I'm Kaela Getz and I'm going to be talking about the results of our paper using the pulse chase assay to track how MHC class 1 molecules are folding and acquiring resistance to endo-H. This experiment yielded a number of supplemental results figures and images, but these are the two critical in answering the initial question. So as mentioned previously, it was thought that these proteins acquired resistance to in vitro treatment of endo-H within the Golgi. Let's take a look at figure A. Now as a reference, the H2D and H2K refer to specific MHC class 1 proteins. HRT refers to endo-H resistant protein and HSD refers to endo-H sensitive protein. Basically, the researchers used pulse chase to capture MHC class 1 proteins at various points at the secretory pathway. Then they visualized these proteins using SES page gel electrophoresis. So what we can see here is that as time goes on, new bands begin to appear. The reason this is happening is because as the proteins pass through the Golgi and become resistant to endo-H, they resist cleavance of their glycans. As a result, the molecular size is different, which is reflected by the varying bands. Additionally, you can see in figure B that the percentage of resistant molecules was calculated and tracked over time. This data was acquired using the results of the gel electrophoresis. Clearly, you can see that the percentage of resistant molecules is increasing over time for both MHC class 1 proteins. This was consistent with the initial idea that these proteins are acquiring resistance at a certain point of the secretory pathway. Furthermore, it supported the hypothesis of the experiment, as these results clearly allowed for the determination of where these proteins are throughout the pathway and whether or not they are being degraded by endo-H. Thank you all for listening.